So in order for a muscle to contract, it first must be signaled to contract. But which organ systems were responsible for signaling in the body? So we saw two. We first had the nervous system, which is very rapid at its ability to send signals. However, its effects are not very long lasting. So that is, when the neurons, which are the signaling cells of the nervous system, were no longer stimulated, then that neuron would also stop signaling the cells that it's communicating with. And the second was that we had the endocrine system, which was slow to begin its signaling process, but it had extremely long-lasting effects, and that was because the hormones that the endocrine system releases will stay in the bloodstream or in the tissue fluid for extensive periods of time and will continue to signal so long as they are present. However, we see that it is the nervous system that controls skeletal muscles. The neurons of the nervous system that specifically control skeletal muscles and stimulate them to contract are called the somatic motor neurons, because the term somatic motor implies skeletal muscle. Cardiac muscle and smooth muscle are controlled to a small degree by the nervous system. However, they are primarily controlled by hormones of the endocrine system, as well as other local conditions that will influence the stimulation and contraction of cardiac muscle and smooth muscle cells. So recall that the cell bodies of neurons are called the neuron somas. And extending from the neuron soma is a very long process that's called the axon. And it's the axon that forms junctions with skeletal muscle cells and will stimulate those skeletal muscle cells to contract. The neuron somas of most of the somatic motor neurons are found in the spinal cord. Some of them are found in the brain, that is for the skeletal muscles of the face and things like that. But these neurons are able to connect from either the brain or the spinal cord to the skeletal muscles that they control by way of very long axons. So sometimes the axons of some of the somatic motor neurons can be as long as 3 feet in length. The junction that is formed between the end of that somatic motor neuron and the skeletal muscle cell that it controls is called the neuromuscular junction. It's also referred to as the motor end plate. So let's take a closer look at the anatomy and the structures of the neuromuscular junction. So the end of the axon at the neuromuscular junction is expanded, and this expanded end is called the axon terminal. And the axon terminal comes in close proximity to, however, it never actually physically touches a structure called the sarcolemma. And the sarcolemma is the plasma membrane of the skeletal muscle cell. So inside of the axon terminal are small vesicles that are called synaptic vesicles. And these vesicles contain a substance called neurotransmitter that will be released from the neuron when that neuron is stimulated. The neurotransmitter will then travel across the very small gap between the neuron and the skeletal muscle cell and will stimulate the skeletal muscle cell to contract. So the neurotransmitter that specifically stimulates skeletal muscle cells is called acetylcholine, which is typically abbreviated ACH. And so in this diagram, we see that these small green spheres that are contained within the synaptic vesicles are the acetylcholine molecules. When released, this acetylcholine will travel across the small fluid-filled gap that exists between the axon terminal and the sarcolemma of the skeletal muscle cell, and it will specifically bind to acetylcholine receptors on the sarcolemma, and it's those acetylcholine receptors that will then stimulate the skeletal muscle to contract. So as mentioned a few moments ago, there's a very small fluid-filled gap that exists between the axon terminal of the motor neuron and the sarcolemma, or the plasma membrane of the skeletal muscle. And that small fluid-filled gap is called the synaptic cleft. The sarcolemma at the synaptic cleft has many receptors, as we just saw, for acetylcholine. And the acetylcholine receptors are what will respond when acetylcholine binds to them, and it will stimulate the skeletal muscle to contract. The sarcolemma is highly folded. These little folds are called junctional folds that help to increase the surface area of the sarcolemma and allow more neurotransmitter receptors to be located at the neuromuscular junction. So let's begin our look at the physiology of muscle contraction. We'll start with the events at the neuromuscular junction. So there are two major events that take place at the neuromuscular junction, and we'll look at the details in a second. The first is that an action potential will be created in the somatic motor neuron, and that action potential will spread along the axon of that neuron to the axon terminal. This action potential will then cause the release of acetylcholine from the axon terminal, which will stimulate the skeletal muscle by binding to those acetylcholine receptors and creating an action potential in the plasma membrane of the skeletal muscle cell, and that action potential will then spread across the entire skeletal muscle cell. So then we'll look at what happens in the skeletal muscle cell after the acetylcholine has stimulated it. So first we see that acetylcholine has to stimulate the skeletal muscle cell and create an action potential in the sarcolemma of that skeletal muscle cell. So take note here that we've seen two action potentials. The first is the action potential occurring in the neuron that's stimulating the skeletal muscle cell. So that's the action potential that's releasing the acetylcholine from that neuron. And the second action potential is the one that's created by the acetylcholine when it stimulates the skeletal muscle cell after that acetylcholine has been released from the neuron. So the action potential that is created in the skeletal muscle cell will go on to cause the release of calcium that's stored in the sarcoplasmic reticulum. The calcium will bind to a regulatory protein called troponin, which is found on the thin myofilament. And when calcium binds to troponin, it causes troponin to change shape, and it moves another protein that's associated with it called tropomyosin away from the myosin binding sites on the thin myofilament. And so this shift in troponin and tropomyosin, when calcium binds to it, will permit the myosin heads to bind to the actin proteins on the thin myofilaments and pull, which is going to shorten the sarcomere. So recall that the plasma membrane of the skeletal muscle cell has these small tunnel-like structures that are called the T-tubules. And on either side of the T-tubule is where the sarcoplasmic reticulum butts up against it. So these expanded ends of the sarcoplasmic reticulum that come in close proximity to these T-tubules are called the terminal cisterns of the sarcoplasmic reticulum, or the terminal cisterns of the SR. So the T-tubules and the terminal cisterns of the sarcoplasmic reticulum are located just above the junction between the A band and the I band within the sarcomere. So the action potential that is created after acetylcholine binds to the acetylcholine receptors on our skeletal muscle cell will spread or propagate away from the neuromuscular junction, and it will spread down the T-tubules. That is because the T-tubules are just these small little tunnel-like extensions of the plasma membrane that go deep into the muscle cell itself. In the walls of the T-tubules, we find small voltage-sensitive proteins, and the action potential is a voltage change in the plasma membrane, and so these voltage-sensitive proteins will respond. And these voltage-sensitive proteins are also connected to calcium channels that are on the terminal cisterns of the sarcoplasmic reticulum. So when the action potential stimulates the voltage-sensitive proteins, they will change shape, and in doing so they will cause the calcium channels on the sarcoplasmic reticulum to open. And this allows the calcium that's stored in the sarcoplasmic reticulum to flood into the sarcomere. and that's where it will be able to get to the thin and the thick filaments. Although specifically we'll see that that calcium functions with some of the proteins that are associated with the thin myofilaments. So when the skeletal muscle is relaxed, we see that myosin is blocked or prevented from binding to actin because of two proteins called troponin and tropomyosin. The troponin supports the tropomyosin, and it's the tropomyosin that's actually blocking the active sites where myosin would bind to actin on. So when calcium floods the sarcomere after the muscle is stimulated, so that is after the action potential is propagated and caused the release of calcium from the sarcoplasmic reticulum, that calcium will bind to the troponin that's associated with a thin myofilament. And that causes troponin to change shape, and in doing so it will move tropomyosin. And when tropomyosin moves, it's going to expose those myosin binding sites on the actin proteins. And so now those myosin proteins on the thick myofilaments are able to attach to actin on the thin myofilaments and pull, which is going to cause the sarcomere to shorten. So when myosin is able to bind to actin, this will begin a sequence that's called the cross-bridge cycle. The cross-bridge refers to the binding of myosin to actin. The cross-bridge is capable of forming so long as calcium is being released from the sarcoplasmic reticulum while the muscle is being stimulated. The second step is referred to as the power stroke because this is where the pulling force is generated. So the myosin, which is now attached to the actin, will bend at its hinge region, which is the little region that's near the myosin heads, and in doing so, it will pull the thin myofilament. At the same time that the bending occurs, ADP and a phosphate ion are released from the ATP binding site on myosin, and these two substances came from an ATP molecule that has been split, and we'll see what that's about in just a second. So the third step is the breaking of the cross bridge, and the cross bridge is broken when a new ATP molecule binds to the ATP binding site on the myosin head. Myosin will then detach itself from the thin filament, so this is going to break the cross bridge. As kind of a side note here, when not long after death occurs, muscles will tend to become stiff and rigid in a condition that's called rigor mortis. The myosin heads are capable of binding to actin and generating tension in the muscles, so even not long after we have died. However, the cells are no longer producing ATP. So the cross bridges will form, so that is, myosin will bind to actin and pull, and the muscles will generate tension. However, the cross bridges cannot be broken because of the lack of ATP, because dead cells do not create ATP. And so this shows us that ATP is an important part of living muscle because ATP is what causes the breaking of the cross bridge so that the myosin can reset itself for another muscle contraction. So the fourth and final step in this is a process called ATP hydrolysis. And this is where ATP is split. And in splitting ATP, the energy that is released from the splitting of ATP will be used to return the myosin heads to their pre-contracted state. This will reset the myosin head for a subsequent contraction, and the cross-bridge cycle after this will repeat itself so long as both calcium and ATP are present. So just to summarize this here, as long as calcium is present in order to expose the myosin binding sites on actin, and so long as ATP is present, which is going to return the myosin to its pre-contraction configuration, as well as break the cross-bridge, this cross-bridge cycle will continue to repeat itself and will continue to generate tension and pulling forces in that sarcomere.